What exactly is a gene therapy? First, let's start with the basics. This is a gene. Genes are made up of DNA, which are the blueprints to build the enzymes or proteins that do the work for your body. As far as we know, humans have between 20,000 and 25,000 genes, and most people have 2 copies of each gene inherited from each of our parents. They are the building blocks of our physical makeup, from the color of our hair, to our athletic abilities, to our immune system. But sometimes genes aren't built correctly. And a small adjustment to the DNA within our genes can change how our proteins work, which can affect how we breathe, walk, or digest food product. If genes are like the blueprint to our body, gene therapy can fill in some missing rights. Gene therapy is the introduction, removal, or change in genetic material, specifically DNA, or its molecule cousin, RNA, into the cell's location to repair or change the gene to treat specific disease. The transferred genetic material then changes how a protein or group of protein is produced by the cell. It can reduce levels of certain disease-causing proteins, increase production of disease-fighting proteins, or even produce new or modified proteins within the cell. How a gene and the genetic material is transferred to the cell during gene therapy. Well, a gene cannot be directly inserted into your cell. Instead, a carrier, also known as a vector, helps deliver the gene. Typically, viruses are used as vectors, because they evolved to be very good at sneaking into and infecting cells. But in this case, their will be is to insert the gene or genes into the cell. The infectious part of the virus is removed, allowing it to still enter the cell's nucleus without making the patient sick. There are 2 kinds of gene therapy: in vivo and ex vivo. Ex vivo is any procedure accomplished outside. In gene therapy clinical trials, cells are modified in a variety of ways to correct the gene. In ex vivo, cells are modified outside the patient's body and the corrected version is transplanted back into the patient. The cells are treated with either a viral or non-viral gene therapy vector carrying the corrected copy of the gene. Opposite of ex vivo is what we call in vivo, where cells are treated inside the patient's body. The corrected copy of the genes is transferred into the body of the patient. The cells may be treated either with a viral or non-viral vector carrying the corrected copy of the gene. If the patient is weak or the cell cannot be extracted out from the body, the gene is introduced directly into the body. Advances in gene therapy research allows to treat and hopefully someday avoid rare and uncurable diseases.